Hello friends, I am Dr. Sonu Yadav. Today's lecture is on metabolism of red blood cell. Now coming on to the introduction part, that is RBCs are anucleate. It does not have nucleus, no nucleus, as well as there are no other organelles. Red blood cell, they made up of mainly cell membrane and cytoplasm. So there are no other cell organelles, there is no mitochondria. And if there is no mitochondria, no respiratory cycle, no citric acid cycle, no oxidation of fatty acids. In short, if there is no mitochondria, there is no energy production, as it is the powerhouse of the cell. So how energy is produced in red blood cell? It is produced from anaerobic glycolysis. That is, ATP is obtained from glycolytic breakdown of glucose with the production of lactate, which is used for keeping the maintaining the biconcave shape, and as well as regulation of transport of ions and water in and out of the red blood cells. Now coming on to the metabolic pathway for the generation of energy, as well as maintaining the biconcave shape and transport of ions and water in and out of the red blood cell. So basically there are two pathways, glycolytic and hexomonophosphate. These are the two mainly metabolic pathways. Glycolytic, this is the main pathway. Only 10% of the glucose enter through hexose monophosphate shunt. It is protective pathway and it is the energy producing pathway. So coming on to the glycolytic pathway. In this pathway, glycolytic pathway, the glucose is converted into pyruvate. It occurs in the cytoplasm of the cell, and as we already know, this is the energy producing pathway. So the net production of ATP during glycolytic pathway is 7. So there is net gain of 7 ATPs, adenosine triphosphate. Now utilization of ATP in RBC. They are used for the phosphorylation of sugars and proteins, for membrane asymmetry, membrane stability, and maintain the red cell shape. So we're discussing about the glycolytic pathway. In this pathway, the glucose is converted into pyruvate. It occurs in the cytoplasm and there is net gain of 7 ATPs. Now what are the steps involved in glycolytic pathway? The site is cytoplasm. Now in this glucose, it enters the red blood cell through a transporter, that is GLUT1. And glucose, it contains 6 carbons. Now this glucose, it is converted into glucose 6-phosphate with the help of an enzyme. Glucose 6-phosphate is converted into fructose 6-phosphate, then fructose 1,6-bisphosphate, and then it leads to production of dihydroxyacetone 3-phosphate and glyceraldehyde 3-phosphate. This, all the first 4 components, they contain 6 carbon, and then dihydroxyacetone, 3-phosphate and glyceraldehyde 3-phosphate, they contain 3 carbons. And dihydroxyacetane 3-phosphate, they are interchangeable. It is converted into glyceraldehyde 3-phosphate. So at the end of this phase, we have 2 molecules of glyceraldehyde 3-phosphate containing 3 carbon. And also remember, this is the energy consumption phase. In this phase, we have utilized 2 ATPs. As the glucose is converted into glucose 6-phosphate, the ATP is converted into ADP. So during this phase, the 2 ATPs are consumed. So this is the energy consumption phase. Glucose is converted by enzyme glucokinase. Then glucokinase will convert glucose into glucose 6-phosphate, and this glucose 6-phosphate converted into fructose 6-phosphate with the help of phosphohexose isomerase. You can memorize it by phospho, 6 carbons, so hexose, and isomerase, because both are isomers. Now fructose 6-phosphate will converted into 1,6-bisphosphate. One more phosphate group is added, that's why it is named as bisphosphate. One phosphate at one position of carbon and one at 6th position. That's why it is known as fructose 1,6-bisphosphate. And the enzyme is phospho and fructokinase. Now this fructose 1,6-bisphosphate is converted into dihydroxyacetone 3-phosphate, glyceraldehyde 3-phosphate. 3 is for 3 carbons. Phosphate, the enzyme is aldolase. So this is the first phase. Now the next is energy producing phase. As we have 2 molecules of glyceraldehyde 3-phosphate containing 3 carbons. This glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, then 3-phosphoglycerate, 2-phosphoglycerate, finally phosphenol pyruvate, and then pyruvate. This is the cycle in which glucose is converted into pyruvate. It has 2 phases, energy consuming and energy producing. So this is the energy producing. In this, when glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate, NADH is formed, and this NADH is equal to 2.5 ATP. And when phosphenol pyruvate is converted into pyruvate, there is production of ATP. As we have 2 molecules of glyceraldehyde 3-phosphate, so there are 2 NADH and 1, 2, 3 and 4 ATP. So 4 ATP, then we will subtract the 2 ATPs as these are consumed during the first phase. So we get 2 ATPs and here 5 ATP. So net gain of energy is 7 ATP. Now just discuss the enzymes. Glyceraldehyde 3-phosphate is converted into 1,3-bisphosphoglycerate. Then this is converted into 3-phosphoglycerate with the help of enzyme phosphoglycerokinase. Then this 3-phosphoglycerate is converted into 2-phosphoglycerate with the help of enzyme phosphoglyceromutase. And then 2-phosphoglycerate is converted into tophosphonol pyruvate with the help of enzyme enolase, and this is finally converted into pyruvate with the help of enzyme pyruvate kinase, and this pyruvate is converted into lactic acid, which is transported to blood and liver. This is all about the glycolytic pathway. So in glycolytic pathway, it takes place in the cytoplasm, and this is the pathway in which glucose is converted into pyruvate, and there is net gain of 7 ATP. There are two phase: energy consumption and energy producing. It starts with glucose having 6 carbons, and this glucose enters into cytosol through a transporter, that is GLUT1. Then this is converted into glucose 6-phosphate, fructose 6-phosphate, fructose 1,6-bisphosphate, and then you have to memorize there are two products: dihydroxyacetone 3-phosphate and glyceraldehyde 3-phosphate. This dihydroxyacetone phosphate is converted to glyceraldehyde 3-phosphate. So at the end of this phase, we have two molecules of glyceraldehyde 3-phosphate. This glyceraldehyde, it went through a series of events and converted into pyruvate with the production of 2 NADH and 4 ATP. Now what is the importance of glycolytic pathway? This is the energy production pathway. There is net gain of 7 ATP. Plus in RBC, 1,3-bisphosphoglycerate binds to hemoglobin, and this will decrease its affinity for oxygen and helps its availability to tissues. So this is the importance of glycolytic pathway. Now coming on to the next pathway, that is pentose phosphate pathway or hexose monophosphate shunt pathway. This is the alternate pathway for glycolysis. Converts hexoses into pentoses, 6 carbon into 5 carbon compound. It takes place in cytosol, and it is the important cycle in generating NADPH. There is no ATP production. No ATP is directly utilized or produced. There is production of NADPH. It has two phases: oxidative phase and non-oxidative phase. Now coming on to the oxidative phase. It starts with glucose 6-phosphate. Now glucose 6-phosphate is converted into 6-phosphogluconolactone. Then this is converted into 6-phosphogluconate, and then ribulose 5-phosphate. This is the product of oxidative phase, end product. So in this, there is production of 2 NADPH, as you can see. This is the oxidative phase. Glucose 6-phosphate is converted into 6-phosphogluconolactone, then 6-phosphogluconate, and then ribulose 5-phosphate. So in this phase, glucose is converted into ribulose 5 phosphate, and there is production of NADPH. This NADPH helps in production of reduced glutathione, that is GSH, and this will help in prevention of oxidation of the red blood cell membrane. So this cycle is also important as NADPH helps in prevention of oxidation of red cell membrane, and it is produced when this glucose is reduced into ribulose 5 phosphate, and these are the enzymes, that is glucose 6 phosphate dehydrogenase, and then gluconoleptonase, then 6 phosphogluconate dehydrogenase, then non-oxidative phase. In this phase, the ribulose 5 phosphate is acted upon by two enzymes, that is epimerase and isomerase, and this ribulose 5 phosphate is converted into ribose 5 phosphate and xylulose 5 phosphate. These two combines and acted upon by transketolase and converted into glyceraldehyde 3 phosphate and pseudoheptalose 7 phosphate. Then both of these combine and acted upon by trans-aldolase and converted into fructose 6 phosphate and erythrose 4 phosphate. Then this combines with xylulose 5 phosphate and converted into fructose 6 phosphate and glyceraldehyde 3 phosphate. Then this is converted into fructose 6 phosphate. So this is the end product of non-oxidative phase, that is fructose 6 phosphate. So during hexose monophosphate shunt, there is production of 2-NADPH, very important, and the glucose 6 phosphate is converted into ribulose 5 phosphate. This is the oxidative phase, and then this ribulose 5 phosphate is converted into fructose 6 phosphate. This is the non-oxidative phase through various series of events. Now what is the importance? This cycle help in fatty acid synthesis, and there is biosynthesis of NADPH used in synthesis of certain amino acids, and it has a free radical scavenging, that is reduced glutathione is regenerated, help in maintaining the erythrocyte membrane integrity as reduced glutathione prevents oxidation of the red blood cell membrane. Now we have discussed the metabolism of red blood cell. As there is no nucleus, no mitochondria, so energy production is through anaerobic glycolysis. Glycolysis means breakdown of glucose. So it is via pentose phosphate pathway or glycolytic pathway. This is the main pathway. In this, the glucose is converted into pyruvate, and this is the ribulose 5 phosphate is the main compound, as there is production of NADPH. and this is the ATP producing pathway. In this pathway, no ATP is consumed or produced. This is all about today's lecture.